Good morning. Today the main topic of this class is about proteins, amino acids, and polypeptides. The essential idea that we must have about this is that proteins have a very wide range of functions in living organisms. Any kind of protein in your body or any kind of living organisms has a specific role. One of the central ideas in biology is that structures dictate function. Above, you can see insulin in its secondary, tertiary, and quaternary structures. Polypeptides vary hugely in the combination and number of amino acids that they are composed from. Even if we consider a single polypeptide, its properties and hence its function would vary greatly depending on its level of structure. Okay? Insulin can exist in all these forms, but the active form, which controls blood glucose levels, is the tertiary structure. This one. So, we have an example of amino acids and how these structures, polypeptides, are formed. We have an example of anabolism by condensation. What is anabolism? The process in which, through the use of bonds, we're going to form new substances using smaller units. Okay, this is amino acid number 1. This is amino acid number 2. How do you recognize an amino acid? Because it has nitrogen in its structure. Okay, so we need a bond to join these two amino acids to form a polypeptide. In this case, this is the bond that is going to join these two structures, and that bond is called peptide bond. This is related to proteins. That is called peptide bond, okay? In which organelle of the cell this process is carried out? In the ribosome. What happens in the ribosome? The ribosome condenses two amino acids into a dipeptide, forming a peptide bond. Everything happens in the ribosomes. The bonds formed are types of covalent bonds. Bonding monomers together creates a polymer. So mono means one, poly means many. This is an example of a protein. You should know that there are 20 different amino acids in polypeptides synthesized on ribosomes. So from those 20 different amino acids, we're going to have a huge range of proteins. Ribosomes are the molecules between cells that facilitate the formation of peptide bonds, and hence where polypeptides are synthesized. So in the ribosomes, we're going to have protein synthesized. Remember that. So how that happened? This is this figure represents the peptide synthesis. When I say peptide, I mean proteins, okay? Because they are going to form different proteins. We have the messenger RNA, the ribosome, and remember the messenger RNA exits the nucleus of the ribosome, the nucleus of the cell, and goes through the ribosome to form peptides. How? We have messenger RNA with information. Remember uracil, guanine, adenine, cytosine, and then we have this tRNA that is transferring RNA that is going to join to the different RNA, messenger RNA, and form the different peptides. So depending on the letters, uracil, is going to form lysine, and for example, adenine, and guanine, that triplet is going to express phenylalanine, and those amino acids that are going to form peptide bonds to form a polypeptide. There are 20 different amino acids in polypeptides synthesized on ribosomes. These are some examples: arginine, histidine, lysine, aspartic acid, glutamic acid, serine, threonine, asparagine, glutamine. So all of these, they have different information. As you can see, they have, if they are amino acids, they must have this C. Remember, this point is a carbon, COOH. It's a carboxyl group, COOH, and also must have nitrogen, NH2, nitrogen and hydrogen, okay, in this way. So as you can see, all of them, they have this configuration. If I ask you in an exam, identify which one is the amino acid. COH, the carboxyl group, and the NH2, that is the amine group. This is one example of an amino acid is called hydroxyproline, and is an example created not by the genetic code but modification after polypeptide formation or proline. So this is one example of how these proteins are expressed. Do I have to learn all of them, the names of all of them and the structure? No, you don't. You just need an awareness of the concepts outlined, okay? And why do we have so many polypeptides, so many different proteins carrying out different functions? Because any protein could be any length. We have 20 amino acids with 20 random formations, and amino acids in any order or combination. So these are the amino acids that can join with different amino acids that you can see. And if a polypeptide contains just 7 amino acids, I have 7 here, there can be 20 elevated by 7 possible polypeptides. In total is 1,280,000 possible polypeptides. I'm sorry, it's 1,280,000,000 possible polypeptides generated. Given that polypeptides can contain up to 30,000 amino acids, the different possible combinations of polypeptides are effectively infinite. That's why we have so many different organisms in this planet. Every single organism in a group of it belongs to a group of species, right? But they look different. Why we have so many humans that they look different? You don't have like a twin. So it's because of this, because the formation of proteins, remember that we have proteins are going to express a phenotype, how do you look like outside, and that is the work of proteins, to form proteins, to form different substances that are going to express different physical characteristics in any kind of living organism. And this is again how I'm going to put a video to show you how this is the mechanism of formation of proteins. I'm going to show you a video. Remember that this happens in the ribosomes. And this is the central dogma of genetics that we're going to study later, but I think that you have the idea because I know that you studied this in biology first. DNA, we have the DNA, then we have the process of transcription and the formation of messenger RNA that happens in the nucleus, that process. The RNA leaves the nucleus and goes into the cytoplasm. And then we have the process of translation to form polypeptides or proteins. Everything happens in the cell. As you can see, DNA, transcription, we have RNA in the nucleus. Then that mRNA exits the nucleus and goes into the cytoplasm, in which the mRNA goes, and thanks to the ribosomes, polypeptide formation is carried out.